The overall goal of this procedure is to image the distribution of metals in a population of cells. This is accomplished by first preparing sterile silicon nitride windows and next growing cells on those windows. Then, the cells are fixed, rinsed into a compatible buffer, and dried. The final step is to image the cells at the beamline. Ultimately, X-ray fluorescence microscopy is used to show the distribution of metals in the cell.
Generally, individuals new to this method will struggle as the windows are very fragile.
Visual demonstration of this method is critical, as growing cells on the windows with minimal cell aggregation, as well as the rinsing steps, are tricky because cells do not adhere well to the X-ray fluorescence substrates.
The first task is to isolate the window. First, using a stereoscope, make sure that a pair of reverse fine-tipped tweezers are straight, aligned, and clean. Next, slightly open the capsule containing the window. Squeeze one end containing the window while rotating the other end. Don't squeeze the window itself. Then, carefully grip the frame with the tweezers while gently squeezing the capsule near the opened end. Apply pressure to make the capsule wider where the window edges should come out and remove the window. Now, prepare some adhesive tape. Place a piece of tape on a clean slide and cut a 1/4-inch strip of tape from the long side of the slide.